Howdy! My name is Olivia Tebow. I am a PhD student here in the Physiology and Toxicology lab, and I work under Dr. Lena Petersen and Dr. David Halla. For Dr. Petersen's work, I work a lot with autoresp and looking at the respirometry of fish. So we measure their oxygen consumption inside of the swim tunnel, and that lets us know their metabolic rate, which we can use to measure their physiology and their physiological fitness. We also have them in a couple of different sizes. We have them in a large size for large like salmonids and trout, and we also have them in a little well for eggs and larvae. For Dr. Halla, I work in endocrinology and toxicology of southern flounder in the Gulf. Southern flounder is declining, and it has been over the past 30 years, and so we want to see if that's due to their reproductive fitness. And so we take plasma samples and we look at a broad spectrum of their hormones and compare that to their current reproductive status. And that lets us know for managers over in TPWD which southern flounder are the best ones to use for broodstock. I got into my PhD because my dad was a professor, and when he passed away when I was in high school, I got a bunch of emails and stuff from other students that he had had in the past and how much he had really affected their lives and changed the way they were going in their career. And that made me want to follow in his footsteps and be a professor as well. I got this position specifically because as an undergrad, I took a lot of Dr. Petersen's courses, and I really liked who she was personally and her research. And so I decided to approach her and did a year of volunteer work in her lab as an undergrad. And during that year, I showed them really good initiative, and I showed up every day on time, and I did really hard work, and they gave me a position as a PhD student the next year. So I went straight from undergrad to PhD, which I would say is 50-50 common here in Texas A&M Galveston, but I know a lot of other students go into their master's before they go into their PhD as well. And it just depends on the opportunities provided to you for those. This is our large swim tunnel. It's for the larger salmonids and trout and other fish like flounder as adults. Water will circulate through, and we will close the system so we can measure the amount of oxygen the fish are using while they're swimming at different speeds in this tunnel. Fish will swim through this large tunnel, and as they swim at different speeds, we can record their oxygen consumption as the water passes this way. We take their minnows and we put them inside the tunnel here, and while they're inside, they'll swim inside the little tunnel and we can record their oxygen consumption. Now, we don't just do this for fish, we can also do it for worms and other small marine animals. This is a microrespirometry system, and we put eggs and larvae into the tiny wells in order to measure their oxygen consumption there.
Hi, my name is Patricia Faulkner, and I'm a PhD candidate here in the Marine Biology Department at TAMA. I work very closely with Dr. Lena Petersen and Dr. David Halla. And my work here as a grad student is very focused on understanding how salinity stress affects the physiology and health of juvenile American alligators. Unlike crocodiles, alligators can't really cope very well with a lot of excess salt in their bodies. We're concerned about saltwater stress in particular because there are a lot of factors that can actually push saltwater into a lot of these freshwater habitats where alligators live. For instance, sea level rise or even hurricanes and storm surges. And we're actually expecting with our changing climate that storm surges and like storms and hurricanes will actually become more frequent and more intense. So we're actually expecting this to be kind of a growing problem. To understand better how saltwater stress affects the health of juvenile alligators, what I've done is run a couple of trials where I would expose a group of juvenile alligators to salt water to compare to a group, like a control group in fresh water. And then I would measure the levels of a lot of steroid hormones, including sex hormones and stress hormones in both of these groups. And what we've found is that not only do the stress hormone levels change in response to this saltwater exposure, saltwater stress, but levels of a lot of sex hormones can change as well. And this is a bit concerning because a lot of sex hormones like testosterone or estradiol play a very important role in how these alligators, juvenile alligators, grow and develop as they're becoming sexually mature. It's important because the more we understand about how this kind of stressor affects their health and their physiology, the more we're able to effectively manage and actually protect alligator populations in the wild. I actually was an undergrad here at TAMUG, and back then we didn't have any kind of succeeding in science course where we learned about all the cool research we do in our labs. And so at the time I was just going to classes, kind of going through the motions, until a friend of mine actually started work on his UGR, or undergrad research project, and he came to me and said, "Pat, do you want to help me research some sharks?" And I said, "Yeah, I want to help you research some sharks." And he was working closely with Dr. Dave Wells at the time, and so I started volunteering and helping him with his project, and before I knew it, I was volunteering with the other labs and just getting all kinds of experience. And just since then, I've fallen in love with research, and I know this is what I want to do. And now I'm actually doing my work on alligators. I've fallen in love with this new animal to study, and that I just love learning about.
Hi, my name is Yu Umeki. I'm a PhD student in Marine Biology department. I'm working with Dr. Halla and Dr. Petersen in Physiology and Toxicology lab. For my dissertation research, I'm studying the metabolic capacity of American alligators. Basically, we are trying to look into whether they can detoxify or metabolize the environmental chemical pollutants in their body, in their system. So the reason why we study this species specifically is that they are very important species along wetland system along the Gulf of Mexico, because they are apex predators. So they tend to bioaccumulate these chemical pollutants in their tissue through the food chain. Also, they are understudied species, which means their physiology or toxicological aspects are not very well known. And also, we are looking into how the wild alligators are affected by chemical pollutants in the wild, in the real world as well.